It feels like practically overnight, we woke up in a world where everyone is suddenly talking about weight loss injections.
Oh, absolutely. They really are everywhere you look.
Right. You see the results absolutely everywhere. I
Yeah.
mean, from Hollywood red carpets where celebrities are stepping out looking completely transformed to, like, the aisles of your local grocery store.
Yeah, where you run into a neighbor you haven't seen in 6 months and they are practically unrecognizable.
Exactly. Drugs like Ozempic, Wegovy, Mounjaro, and Zepbound, they have just saturated our culture to the point where they're basically just part of everyday conversation now.
They really are.
But beneath all those flashy headlines and the dramatic before and after photos, there is this glaring unspoken mystery.
The variance in how people actually respond to them.
Right. Because for all the incredible success stories you hear about, there is a hidden reality. Here is a strange question for you. Why does the exact same dose of the exact same medication act like an absolute miracle for one person, melting away weight effortlessly, while another person taking that identical shot barely loses an ounce?
And instead, they just feel horribly, debilitatingly sick.
Yes. Why does that happen? Well, welcome to the Deep Dive Lab. Today we are looking at a brand new genetic study that proves this wildly unpredictable experience isn't just random chance.
No, it's not random at all.
Scientists have finally found the hidden blueprint that answers this question, and it has been buried deep inside our DNA this entire time. Okay, let's unpack this, because this is a huge deal, right?
It's massively important. I mean, it is one of the most profound contradictions in modern medicine. We are looking at a class of drugs that has fundamentally shifted how we approach metabolism and obesity. By now, most people know the basics of how these GLP-1 medications work. They mimic natural hormones to suppress your appetite and slow down how fast your stomach empties.
Right, they tell your brain you're full.
Exactly. But the individual experience of actually taking them has remained just bafflingly inconsistent. For doctors, prescribing them has been a little like throwing a dart while blindfolded.
I can imagine. Because the data we've seen on the variants is just wild. Like, I'll hear the headline statistic that people lose about 10 to 11% of their body weight on average.
Right, the average.
But averages are so deceiving, right?
Yeah.
Because they completely smooth out the extremes. I was looking at the clinical data for semaglutide before we sat down, and nearly 5% of patients achieved over a 25% reduction in their body weight.
Which is life-altering. That is phenomenal efficacy.
It really is. But then, on the flip side of that exact same study, a massive 32.2% of people achieve less than a 5% weight reduction.
Some of them even gained weight.
Yeah. It's like buying this highly rated, super expensive coffee maker that randomly brews a perfect barista-level espresso for you. But when your neighbor buys the exact same machine, it just pours cold tap water into their mug.
That is a great way to put it. And that variance was incredibly frustrating for researchers.
So what did they think was causing it? Did they think people were just, like, eating differently?
Initially, yeah. The scientific community assumed the answer had to lie in non-genetic factors. So they looked at behavioral and demographic patterns first.
Okay. Like what?
Well, the clinical patterns showed that women tended to lose slightly more weight than men on these medications, around a 12.2% drop for women compared to a 10% drop for men.
Oh, interesting.
Yeah. They also noted that individuals who were taking the medications specifically to manage type 2 diabetes lost significantly less weight than those taking it solely for obesity.
Okay, so the assumption was that factors like your age, biological sex, starting BMI, and whether or not you have diabetes were driving the bus.
Right. They do drive the bus, but it turns out they're only steering it a fraction of the time.
What do you mean?
Well, when biostatisticians built a comprehensive model combining all of those non-genetic factors, age, sex, starting BMI, the exact drug dosage, all of it, that entire model only explained about 21.4% of the variance in weight loss.
Yeah. Wait, only 21%?
Just 21.4%.
Meaning nearly 80% of the reason why the drug works like magic for you and does absolutely nothing for me was completely unaccounted for.
Exactly. A huge blind spot.
So how did scientists figure this out? Because this is where the story gets really interesting. Somebody finally decided to look at the genetic level, right?
They did.
Yeah, I was reading this study that just came out in the journal Nature on April 8th, 2026. Lead researchers Dr. Kiawan Jane Su, Dr. Adam Otten, and the 23andMe research team tackled this missing 80%.
And what's crazy is the sheer scale of how they did it.
Right? They didn't just use a few hundred clinical trial patients. They analyzed a massive data set of 27,885 people who were actively using these GLP-1 medications.
And that scale is precisely why this discovery is so groundbreaking. Traditional clinical trials, which usually only have, a few thousand participants at most, they simply do not possess the statistical power required to find tiny, subtle genetic signals.
Because it's like finding a needle in a haystack.
Exactly. To find a needle in a genetic haystack, you need an unimaginably massive haystack.
Makes sense.
So the researchers achieved this by combining self-reported survey data from the 23andMe research participants with electronic health records, or EHR, which were pulled directly from Apple HealthKit.
Okay, wait. I have to pause you right there.
Sure.
Because looking at the methodology, there's a huge gap in those numbers. Like, the self-reported BMI drops from the surveys were dramatically larger than what the electronic health records show.
Right, yes, they were.
People self-reported an average drop of 11.8% of their body weight. But the actual verified electronic health records showed an average drop of only 5.79%.
That is correct.
So, human nature being what it is, it looks like we are all a little generous with our own success stories. Don't people just lie about their weight on surveys? Doesn't that discrepancy totally compromise the data?
It is a totally fair critique. Honestly, it's the first thing any data scientist would flag. The absolute numbers were indeed different.
Right.
And that's likely due to a combination of self-reporting bias, like you said, and the fact that the electronic health record cohort was skewed slightly younger and had a higher percentage of men.
Ah, okay.
However, when the researchers ran the statistical models, the underlying trends correlated beautifully.
Really? Even with the exaggerated numbers?
Yes. The people who reported the most relative weight loss in the surveys were the exact same genetic profiles showing the most relative weight loss in their official medical records.
Oh, wow. Okay.
The absolute pound shed might have been exaggerated in the surveys, but the biological signal of who succeeded and who didn't was perfectly aligned across both data sets.
That makes a lot of sense. So as long as the trend lines match up, you can still hunt down the genetic culprit.
Exactly.
And to do that, the paper says they conducted a genome-wide association study, or a GWAS. Now, I know this term gets thrown around a lot in modern biology, but can you break down what that actually looks like in practice?
Sure. Think of a GWAS as a massive computational search operation across the entire human genome.
Okay.
You take the genetic code of all 27,000 people, and you have algorithms scan through millions of tiny genetic variations.
Just looking for patterns.
Right. You are looking for a specific variation that appears far more frequently in the people who lost a massive amount of weight and is largely absent in the people who lost very little.
Got it. And what did they find?
When they ran this scan, the algorithms flagged a literal spelling mistake in a gene called GLP-1R.
GLP-1R. So that stands for the glucagon-like peptide-1 receptor gene. That is the literal target the drug is aiming for, right?
It is the bullseye. The specific variant they found, labeled RS1030542, is what geneticists call a missense variant.
Okay, let's define that. A missense variant means the DNA code is mutated just enough to swap out one specific building block, like an amino acid, for a different one when the body is building that receptor.
Precisely.
All right.
In a standard GLP-1 receptor, the seventh amino acid in its signal peptide sequence is supposed to be proline.
Proline, okay.
But in people who carry the specific genetic variant, that proline is swapped out for an amino acid called leucine.
So it's just a tiny chemical swap, proline to leucine. How does something so incredibly microscopic change whether a person loses 30 pounds or 0 pounds?
To understand the mechanism, we really have to look at what a signal peptide actually does.
Okay.
You can think of the signal peptide as a molecular barcode or a shipping label. It's attached to the receptor when it is first being manufactured deep inside the cell.
Like an internal delivery system.
Exactly. It tells the cell's internal machinery, hey, transport this receptor to the outer surface of the cell membrane so it can do its job.
Okay, so if the receptor is a package that needs to be delivered to the surface of your cell so the medication can find it in your bloodstream, what does upgrading the shipping label to leucine actually do?
Well, the leucine amino acid is physically much more hydrophobic than proline.
Meaning it repels water.
Right. It actively repels water and is highly lipid soluble. When the cellular machinery is trafficking this receptor package to the surface, it has to pass through and embed itself into various lipid-based membranes.
Oh, I see where this is going.
Because leucine is more hydrophobic, it embeds into those membranes much more efficiently. It makes the entire transport process far more stable.
So more of them actually survive the trip.
Exactly. The result is that a person with this leucine variant simply ends up with a significantly higher density of these receptors successfully delivered to the surface of their cells.
So they literally have more molecular catcher's mitts on the surface of their cells, ready and waiting to catch the drug when it enters the bloodstream.
Yes, it doesn't necessarily change how tightly the drug binds to the receptor, but purely by having a higher population of functional receptors available, the cellular response to the drug is powerfully amplified.
Wow. Okay, I love the biology of that. But I do have to push back a little on the actual real-world impact of this specific variant.
Fair enough.
Because researchers noted that having this specific T allele, this upgraded shipping label, results in an additional 0.76 kilograms of weight loss per copy of the gene. That's only about 1.7 pounds.
Right.
And since we get one copy from our mom and one from our dad, even if you hit the genetic jackpot and inherit the variant from both parents, you're looking at maybe 3.5 pounds of extra weight loss.
That's true.
In the grand scheme of someone trying to lose 50 or 60 pounds, is a 3.5-pound difference really a game changer? It sounds like a biological footnote.
It's a highly perceptive question, and on its face, 3.5 pounds might not sound revolutionary, but you have to view this through the lens of compounding effects and medication tolerance.
How so?
That 1.7 pounds per allele is an isolated baseline effect. It proves that the biological efficiency of the drug is fundamentally tied to the structural stability of the receptor encoded by your DNA.
Right.
But more importantly, this higher receptor density changes the trajectory of a patient's entire treatment plan. If your cells are highly receptive, you might achieve your target weight loss at a much lower dosage of the drug.
Oh, wow. I didn't think about that.
Yeah, that means paying less money and critically minimizing the long-term stress on your gastrointestinal system. And that actually brings us directly to the second, arguably more profound revelation of this study.
Right, because if the drug is working better at the cellular level, like if you have all these extra receptors, catching the medication and amplifying the signal, does that mean you feel the side effects more too?
Yeah.
Because these drugs are notorious for making people feel completely sick to their stomachs.
What's fascinating here is when the researchers ran a second genome-wide association study, this time focusing entirely on the severe side effects people reported, the data confirmed exactly that.
Really?
They found that the exact same genetic signals located in the GLP-1R locus that were linked to more weight loss were also highly correlated with increased nausea and vomiting.
Oh, man. Okay, if you are listening to this right now and you've been miserable on one of these drugs like feeling sick every morning, dreading your next meal, this data is actually saying that your misery is proof that the drug is binding perfectly to your cells.
It is a biological double-edged sword. Your pain is literally a signal of peak efficacy. The better the drug works, the rougher the ride.
That is so crazy.
And to prove this wasn't just a coincidence, the researchers used something called colocalization analysis.
Colocalization analysis. Okay, break that down for us.
Imagine you have two transparent maps of a city. One map shows all the hot spots for traffic accidents, and the other map shows all the locations of poorly timed traffic lights.
Okay.
If you lay the transparent maps on top of each other, and the hotspots align perfectly on the exact same GPS coordinates, you have colocalization.
They overlap perfectly.
Right. In this study, the researchers mapped the genetic hotspots for maximum weight loss, and they mapped the genetic hotspots for severe nausea. When they overlaid them, the signals originated from the exact same genetic coordinates.
Wow.
The individuals whose bodies were most receptive to the drug's intended effects, suppressing appetite and slowing gastric emptying, were concurrently experiencing the most intense adverse gastrointestinal effects.
That is wild. It totally reframes how we should think about the side effects. It's not just your body, like, rejecting the drug.
Yeah.
It's your body being almost overwhelmingly responsive to it.
Exactly.
But wait, there was a second genetic twist in this study involving a different gene entirely, right?
Yeah.
And this one seems specific to only certain brands of the medication.
Yes. And this is where the science transitions into direct, personalized medicine.
Tell me more.
As we touched on earlier, drugs like Wegovy and Ozempic use semaglutide, which targets only the GLP-1 receptor. It is a single agonist drug.
Okay.
But drugs like Mounjaro and Zepbound use a compound called tirzepatide. Tirzepatide is a dual agonist. It targets the GLP-1 receptor, but it also heavily targets a second different receptor called the GIP receptor.
So it's hitting two different buttons in the body's metabolic control panel instead of just one.
Correct. Knowing this, the researchers decided to look exclusively at the population of people taking tirzepatide, and they discovered a second, highly impactful genetic variant, this time in the GIPR gene.
Which is the gene responsible for building that second receptor.
Right. This specific variant is called RS1800437.
And the paper notes that if a person carries the mutated C allele in this GIPR gene, their risk of experiencing moderate to severe vomiting while taking tirzepatide jumps by almost 1.8 times.
Almost double the risk of severe vomiting.
That is brutal. But let's dig into the why here. If the first gene we talked about makes the drug work better, what is this second gene doing to cause so much sickness?
To understand the mechanism, we have to look at how these two different neuronal systems interact in the brain and the gut.
Okay.
The GLP-1 side of the drug is what primarily drives the nausea. It sends powerful signals to the emetic center of the brainstem, the area postrema, and forces the smooth muscles of the stomach to drastically slow down their contractions.
Which makes you feel sick.
Right. However, preclinical studies have shown that activating the GIP receptor actually acts as a physiological buffer.
A buffer, yes.
When GIP is activated, it modulates overlapping neuronal pathways to essentially raise the threshold for nausea. It sends competing, calming signals to the brainstem and helps regulate those stomach contractions, naturally attenuating the aversive effects of the GLP-1 activation.
So the GIP side of the drug acts like the shock absorbers on a car, actively smoothing out the bumpy, nauseating ride caused by the GLP-1 side of the drug.
That is brilliant. Yes, and taking that shock absorber analogy a step further, the specific genetic mutation they found, that C allele, is a partial loss-of-function mutation.
Meaning it doesn't work right.
Exactly. It changes the 354th amino acid in the GIP receptor from glutamic acid to glutamine. This structural change severely impairs the receptor's ability to transmit its signal.
So it essentially breaks the shock absorbers.
It breaks the shock absorbers. If you have this mutation, the GIP component of the tirzepatide medication can no longer effectively send those calming signals to the brainstem. The protective buffering mechanism is diminished, leaving your body fully exposed to the severe vomiting side effect triggered by the GLP-1 activation.
So what does this all actually mean for the listener? I mean, it means your DNA is having a highly specific chemical handshake with the exact brand of medication you chose.
It does.
It's no longer just a question of, do these weight loss drugs work for me? It's, does my specific genetic code prefer the single-action drug or the dual-action drug?
If we connect this to the bigger picture, what we are truly talking about here is the end of the one-size-fits-all era and the dawn of precision medicine in the realm of metabolic treatment.
Which is long overdue.
Definitely. Currently, prescribing these incretin medications is largely a guessing game based on clinical averages. A doctor prescribes a starting dose of whatever drug is covered by insurance, monitors the patient for a few months, and if the side effects are too severe or the weight loss is inadequate, they might blindly adjust the dose or try a different brand.
It's just entirely trial and error.
Yes.
Which takes months of feeling sick or months of paying for an incredibly expensive drug that isn't doing anything just to find out it was the wrong biological fit from day one.
Exactly. But imagine the healthcare landscape of the very near future.
Oh.
Instead of trial and error, a physician could review your genetic profile and say, hey, you have the GLP-1R variant for high efficacy, meaning your cells have a high density of receptors. This drug will work very well for you.
That would be amazing.
But they could also say, however, you also have the GIPR loss-of-function variant that eliminates your body's natural nausea buffer. Therefore, we know the dual agonist tirzepatide will make you violently ill. Let's avoid Mounjaro entirely and start you on the single agonist semaglutide instead.
That is so specific.
And because your cellular receptors are highly efficient, we can start you on a much lower baseline dose to minimize any standard nausea while still achieving your target weight loss.
Wow, that completely revolutionizes the patient experience. It takes the frustrating guesswork completely out of the equation.
It really does.
But it also makes me think about how this fundamentally changes the way we view obesity and weight loss as a society.
Yeah.
I mean, for decades, there has been this horrible, pervasive stigma that weight loss is purely a matter of willpower.
Right. The old eat less, move more mindset.
Exactly. If you can't do it, it's a moral failure. And even now, with these incredible blockbuster drugs, if someone takes Ozempic and doesn't lose a pound, people might secretly judge them and think, wow, their diet must be absolutely terrible if even the miracle drug can't help them.
And that stigma is not only incredibly damaging, but this research provides direct, irrefutable biological evidence to dismantle it completely.
Well, I love that.
When a GLP-1 drug fails to produce results for someone, it is not a failure of their willpower, and it is not necessarily a failure of their diet. It is a literal, mechanistic, genetic mismatch between the molecular structure of the medication and the cellular receptors encoded by their DNA.
It's just biology.
This study proves that our bodies are not identical metabolic machines. The way we process hormones, the way our brainstem interprets nausea, and the way our cells physically respond to pharmacological interventions, all of that is fundamentally hardwired into our unique genetic code.
It's so validating for anyone who has ever struggled on these medications. It shifts the blame entirely away from the patient and puts the focus back on the molecular biology where it belongs.
It really is a paradigm shift.
Okay, well, we have covered a massive amount of groundbreaking science today. Let's summarize the key takeaways from this deep dive so we can all wrap our heads around what this means moving forward.
Certainly. First, we now understand that your specific DNA, particularly tiny amino acid variations in the GLP-1R and GIPR genes, drastically alters both the volume of weight you will lose and the severity of the sickness you will feel on these medications.
Right. The genes matter.
Second, experiencing brutal gastrointestinal side effects like nausea is actually strongly correlated with the drug being highly effective in your body. Your sickness is an indicator of cellular binding efficiency.
So pain equals progress, biologically speaking.
In this very specific case, yes. And third, this 23andMe and Nature research marks our definitive entry into an era of precision weight loss medicine, where your genetic code will dictate exactly which specific molecular compound and dosage you should be prescribed.
It really is a revolution in how we understand the complex chemistry of our own bodies. And it leaves me with one final fascinating thought to ponder. If a tiny genetic variation, just a single spelling mistake, changing a proline to a leucine on a microscopic cellular shipping label, can dictate whether a blockbuster weight loss drug transforms your body or just makes you violently ill.
Which is mind-blowing.
Right. So what other everyday medications are playing out a hidden genetic lottery inside you right now?
Oh, that's a great question.
From the basic painkillers sitting in your bathroom cabinet, to your seasonal allergy meds, to antidepressants, how many of our daily struggles with medication efficacy and side effects are just waiting for a genetic map to show us the way?
The science is definitely heading there.
Well, thank you so much for joining us in the Deep Dive Lab today. We hope you enjoyed this journey into the DNA of weight loss and the future of personalized medicine. Please be sure to follow and subscribe for more fascinating science stories unfolding in real time. We'll see you on the next Deep Dive.